Yeah, so this is physics. I'm going to do the thing they do in airlines, right? This is physics 354, 454. You're supposed to be here, keep sitting. If you're not, you're free to keep sitting, but you're in the wrong place. All right, so today what I thought I'd do is sort of give you an introduction to the field. So to just give you an idea of the kinds of questions and the kinds of concepts that people like us worry about. And then we'll do I'll go over some of the mechanics of the class, so some of the nitty-gritty, whatever questions I'm sure you have about how the class will actually work. And then we'll do a little bit of intro into some of the material that we need to all understand before we get really started into analyzing some of these systems. So biophysics is a pretty, it's, even though it's sort of a branch of either physics or biology, however you want to look at it, it itself is a giant subfield of both of these disciplines. And not only that, the discipline itself is currently undergoing really sort of dramatic transformations in the kinds of questions that we can address and the kinds of systems that we look at and the complexity of those systems. So to give you a little bit of an overview into sort of how we think about, where different, even sub-branches of biophysics fit in. What I thought I'd do is go over a sort of very oversimplified schematic plot of, where different kinds of questions are thought about. So this plot has two dimensions. So this is a physics class, so we like to think about things, along quantitative dimensions. And so the two dimensions are, on the y-axis, we have the number of components that we're going to look at in a given problem that we want to study. Okay? And this number can range dramatically, right? So you can have everywhere from single component systems, right? So one thing at a time that we really want to analyze the data, basically. Or you could have up to tens, hundreds, even thousands of components interact, right? The whole cell has millions of things interacting with each other. That's one dimension along which we can think about different problems. The x-axis is a length scale over which we want to examine those components. And this also can range very dramatically, everywhere from giant kinds of things, kilometer scale, to hundreds of kilometer scale things, like ecological and evolutionary kind of time scale questions, which is something that is very, very actively worked on actually, even in physics departments. All the way down to nanoscale kind of processes, right? So this is atomic molecular scale events that drive cellular behavior. And everything in between is worked on. So to give you an idea of where this kind of work got started, you can imagine, what quadrant of this do you think we got started with? What would your guess be? So,
like macro scale?
Yeah, right. So kind of not too small because that's really hard. Not too big because that's also hard. Like satellites or something. So somewhere around here probably, right? And what about on this axis? Yeah, kind of lower, right? Yeah, we don't want to be studying thousands of things, even if they are like a sort of meter big. That also becomes very hard. So basically, the sort of traditional starting point for biophysics was in this area of so-called medical physics and biophysics, right? So this is stuff like what you go to the hospital to actually even use today, right? So you can do things like echocardiograms, right, where we have quantitative information about things like the dynamics of blood flow through vessels, right? So these are things you can see with your own eyes unaided, right? And there are only a couple of them that you look at once, right? So you're not trying to necessarily image the entirety of your body all the time, although there are applications like that too. You're trying to really analyze sort of like what is your heart doing and how is it going wrong, right? So the number of components we think about in this scale is relatively limited, and they tend to be things that we can see with our own eyes unaided. Okay. Then people got really interested in applying some techniques that had been pioneered to look at really, really small things. Right? So you can go all the way down to these sort of, again, molecular scale processes with really fancy microscopes. But there, to get started, also people look at small numbers of components, right? So if you want to sort of look up the kinds of work done in this area, this really goes under the name of sort of single molecule biophysics. So this is examining the molecular behavior of individual molecules that are responsible for really important biochemical reactions, for example. So for, one example is, actually I should turn the slide off. So for example, what's shown here are these green blobby things, right? So what are those green blobby things? They are individual molecules of a motor protein called myosin 6. And myosin 6 is a very important player in trafficking cargoes and other cargoes and sort of mesoscopic scale structures around the cell. They track along, it's kind of hard to see in this image, they track along these red fibers called actin. Okay? And so what you can do is you can actually literally watch, I don't know if I have a, yeah, I do have a video, okay, so you can literally watch individual molecules of myosin 6 and study their motion, actually, right? Their actual molecular scale motion to see how that really works, right? How does it actually achieve this function of transporting cargo from one part of a cell to another? So let's take a look at what this looks like. You can sort of watch it, walking along. It's a pretty short movie because these are hard to, these are very difficult data sets to obtain. Okay. Let's see it one more time. So you can see it. So by doing this, you could actually test quantitative models of the kinds of motion that these individual myosin molecules are undergoing, right? You can imagine that at this scale, this is really, really tiny, right? Molecular scale stuff. Even like water colliding with myosin molecules is going to cause a huge impact, right? So you'll have potentially diffusive motion, but at the same time, these myosin molecules are burning energy to actually affect their net transport, so you can have more energetic-driven motion. And the idea is that you can actually propose models for the different kinds of transport and test it against this kind of quantitative data that you can take off of single molecule imaging. Okay, so at another scale is where you can have really big things, but lots of them. And this is the scale of sort of population dynamics, evolution, ecology. So you can study things like how do species interact to grow and to be in a sort of evolutionary, in an ecological niche. You can study how the dynamics of mutations and how mutations spread through population. That's a very important and interesting problem that a lot of people work on. So to give you an example of this, what you have here are two strains of E. coli, these are bacterial cells, that are each labeled in different colors. Okay, the top set of cells are labeled in green, and the bottom set of cells are labeled in red. And in this movie, what they were interested in is just to see how you can infer things about the population dynamics by looking at the interface of these two growing colonies. Okay, and then the fluctuations in that interface can allow you to test, again, very quantitative models of how growth works, how growth in a population works. So there's an example. You can see they're growing out, and they start colliding into each other. It's like two galaxies colliding, right? And you can see that at the interface of these two, you get these kind of ridges and fingers coming out. You can actually analyze those kinds of data to see, what kind of models can explain the fluctuations in that interface, right? How does that constrain models of population dynamics? So in this class, we're going to work kind of in this sweet spot in the middle. Okay? So we're going to mostly focus on on processes that occur at this sort of cellular length scale. So this is at the micron, maybe up to a millimeter, and down to even sort of hundreds of nanometers level scale. And the kinds of systems we'll look at will have a reasonable amount of components, not thousands, not really even hundreds. This is kind of a little bit of a lie here. It'll be, at a scale where we can reasonably do either analytic pen and paper theory and actually try and write down simple models and analyze them with our own hands, or at least sort of stripped down computational models that that won't sort of take the age of the universe to run, right? So, something where we can understand based at a sort of small scale. And so that's what I'm going to define as where we're going to work in this class. So what is this field of systems biology or quantitative biology? Or it goes by, yeah, it seems like there are more names for it now than there are papers sometimes. So today, for this class, we're going to take a very kind of functional, like, sort of operational definition of what systems biology is. And what I'll claim is that systems biology is the study of how biological components interact. So it's not so much the study of the components themselves, but how they interact with each other to produce some definable function. Okay? So there's going to be a connection between the interaction of molecules and the sort of engineering or human discoverable kind of function that they're trying to achieve. So there's going to be a lot. So in this field, there's a lot of thinking behind trying to identify things like what does it mean to have a function, right? This is unlike purely non-living physical systems where there's really no notion of function, right? What's the function of the pressure of water or a pressure of air, right? It's not even a meaningful question in some sense, right? But in evolving living systems, there really is a notion of function, right? These cells, if a dinosaur comes after you, you need to respond to it somehow, right? You can't just sit there, or else you won't be living much longer. So you need to. So no matter what system we're going to look at, we're always in the back of our mind. We should keep this notion of function. Like, how does this system actually achieve something that the cell is trying to do? And what's nice about this scale of working and this definition of systems biology is that a lot of the research in systems biology is rooted in this sort of classical physics tradition of trying to identify the simplest set of assumptions and components and interactions that you need to create a mathematical model of the system that still is able to capture the essence of the function that you're trying to analyze in the cell. So people who work in this area tend not to necessarily focus so much on identifying every single component that that impinges on a given function, right? There's often always something else that we don't know about that affects whatever it is we're trying to do. But we at least try to strip down the system enough so we can rationalize how these interactions give rise to function, and then this kind of work can often either tell us like, oh yeah, we basically described the essence of this function, or it's like, this model doesn't work at all, and there's clearly some other factor, there's some other molecule in the cell that we're missing knowledge about, that we need to discover in order to move forward still. Right? So using this very quantitative and explicit framework in terms of really laying down like what your assumptions are and quantifying what you think those assumptions end up giving you is just a really nice way to even figure out how much you even think you know, right? I mean, in biology, it's so complicated. It's often really hard to even figure out what you know, or you think you might know something, but actually you might not know as much as you think. All right, so let's take a simple example of how these kinds of principles play out in the analysis of a really important biological system. So how do things interact to define a function, and how can we maybe approach this mathematically? And so the system that let's introduce today is the one of chemotaxis. Anyone know what chemotaxis is? Anyone want to define it for us? Remember at all? Yeah?
So it's like the sensing of chemicals and like making movement.
Exactly, yeah. So there's all kinds of N-taxis kind of processes, right? There's phototaxis where like plants will grow towards the sunlight, right? Have you ever seen this? It's really kind of creepy. And chemotaxis is the process by which cells will find chemical gradients. There's gravitotaxis, so there are actually organisms that will find follow the gravitational field. Magnetotaxis, birds do this. They find true north and know where to go. So chemotaxis is the process by which cells follow chemical rate. This is a major thing that cells do, right? Because they're constantly in interaction with their chemical environment. So this is a classic movie of the process of chemotaxis. This is probably what's happening inside my body right now, if you can tell, hopefully. So this is a bacterial cell in that little black image. And I'll play this movie, and this is a movie of this neutrophil cell. That's what this big guy is. So that's an immune cell in the system in our bodies. And you're going to watch it chase this bacteria. Okay? And there's sort of an aesthetic, pleasure from just watching this, but also you should think about this is a cell. This is not like doesn't have a brain or something. It doesn't have free will that way, right? But somehow through these molecular computations, it can do this amazing feat. So here we go. Bacteria, neutrophil, there's a red blood cell. Here they go. So, right? No brain, no like neural system at all, but it's really sort of chasing this guy around, right? It's like pushing other guys away. It has to somehow know that it's like there's other stuff there that needs to, and then it finally engulfs it at the end. But also, it's not perfect, right? That's the other thing to remember. It didn't just gobble it right away, right? It needed to chase it for a little while. And, right, this bacteria, you can watch it again. This bacteria is actually relatively straightforward for the neutrophil to catch because it doesn't appear to have any kind of active, like flagellar kind of, it's not escaping through some sort of active transport. It really looks this motion looks very diffusive. In fact, to be honest, a lot of people speculate this is actually dead bacteria. It's not alive, but I'm not sure. So it has to integrate this chemical cue that the bacteria is shedding with the mechanical ability of the cell to propel itself toward the bacterium. And these mechanical things, it has to push these red blood cells aside that are in the way, right? So it has to integrate all the information, all without a brain, right? No brains. So how in the world does a single cell use molecular scale processes to perform these kind of computations? That's really one of the things that we want to really understand. So, chemotaxis. So what are the kind of questions? What kind of questions would you want to answer if you were? So you can ask questions like, what are the molecules that the neutrophil is sensing? That's a very reasonable question. Or what are the actual molecules that bind that ligand, right? That actually bind the molecules that the bacteria is shedding. Totally reasonable questions, but let's leave those to the side, okay? And let's focus purely on sort of treating the neutrophil as a device. Okay? It's a device that needs to chase bacteria and eat it. So what kind of questions would you want to ask? If you wanted to sort of make some insight into this, what do you think? Speculate, go ahead.
I was wondering,
go ahead, yeah.
I was wondering when the mutation occurred that allowed a cell to chase.
Okay.
And did it occur before, or like how many other mutations, how many other features came before and after this feature?
Okay, that's a good question. That's evolutionarily very sort of a profound question, right? Now that question might not necessarily help you how this one is actually being
Yeah, right.
Okay, yeah, go ahead.
Where and is there any form of depth to the chemical receptors? Are they located across all of the cell? Is there any depth?
Very good. That's a really good question, right? Is there some pattern on the cell surface itself that is sort of sensitive to these? Yeah.
My question was when you're asking these types of questions, what's the balance between more physical related questions like modeling its behavior as a particle or more biology related questions
Yeah, so that to be honest, day to day, that's the hardest question that we ask, right? It's sort of even what exactly are we trying to ask? Yeah, right. Let's assume that to start we want to ask a question of the sort of the biological principle under which this is working, or it could be biophysical. Whatever that. And then we can translate that into sort of a mathematical framework and ask questions of how to actually analyze that, that picture that we're trying to develop. Okay, yeah.
What signaling cascade or what process kind of links the chemical to the mechanical?
Very good. Yeah, that's another really good. Yeah, and even sort of to build on that, basically how does that transduction of that signal even work sort of in terms of, right? So the bacteria, it's not following a straight line path, something that the cell can kind of determine the compute and go to it, right? It's like it's randomly diffusing. So somehow it has to know the current situation in terms of where the bacterium is, go towards it, but it has to be able to update that, right? So that signaling interplay between the sensing of the molecule and its actuation in terms of like where to reorient the cell is a very important question, and that's the kind of that's very much the kind of question we want to answer. So yeah, these are all really good ideas. Yeah. So basically, you got pretty much all the ones that I would have wanted to answer, right? So how not in the sense of molecular identity, but in terms of like binding and unbinding kinetics of this molecule. How does the neutrophil actually sense where the bacterium is? How does it coordinate sensing and responding via motion? This is the last thing we were just discussing. And also importantly, how does it know when to stop, right? These systems, we always we naturally think about how they actively are responding to a certain stimulus or environmental condition, but these are energetically expensive processes, right? When the cell is moving around like that, it's burning energy, right? And this is all stuff from what we eat, basically. So how does it know when to stop so that it stops burning that energy? And then you can ask slightly more, okay, so those are sort of device level questions about this particular system in terms of like how it actually operates. But then you can ask more abstract questions also that move more towards a sort of a pure physical understanding of the constraints on this system, right? So you can ask things like, okay, I have a bacteria, I have some sort of sensor on my bacterium. It has to measure the concentration of some molecule the bacterium is shedding, right? Are there any limits to how sensitive a cell can even be to that concentration profile? Can you build a signaling system that is absolutely perfect, like that it will respond to even a single molecule that it sees? And if you try to build a system like that, will it be sort of hypersensitive, right? Like, will it also respond a lot to noise, basically, right? Will it turn on even in the absence of an input from an actual bacterium? Right? And how do you balance sensitivity and the ability to turn on? So you can ask those kind of physical limit questions, and then you can actually ask things like, you can actually take data from movies like this and actually actually compare these kind of theoretical ideas to real data and see how close does a real cell, given that it's evolved under evolutionary pressures for billions of years in some cases, how close does that cell actually come to achieving these kinds of performance limits that are set purely by physical mechanisms? So let's actually go ahead and unpack this a little bit. Okay? Let's actually try and see if we can get some insight into how to go about not just posing these questions, but actually solving them. Right? That's actually what we're in the business of doing, is posing questions and solving. So, how does the neutrophil sense where the bacterium is? And we had some discussion about, maybe the receptors for the ligand that the bacteria is shedding, maybe they themselves already build a kind of internal map of where the bacterium is. That's one very reasonable possibility. In fact, that's in this particular system, that's very likely how it works. How do we go about actually formally testing that idea? So what are the important players in this system? So we're going to take this very, very stripped down. Obviously, this is a very complicated cell. It has lots of things in it. It has DNA. It has proteins that do other things. It has metabolism. Like it has to be making ATP. We're going to ignore all of that right now. Okay? We're just going to focus in on how does the cell sense this bacterium, and we're going to focus on just this minimal components we need to actually pose that question sort of rigorously. So there are two things that we need to consider, right? And here there's the bacterium itself, which is going to shed molecules that the cell needs to sense. And in green dots here, I've basically written down, or I've cartoonized the sensors themselves. Okay? And again, in the spirit of being as minimally assumption as possible, let's assume for now that these receptors just sort of evenly coat the surface of the cell, okay, and see how far just something like this can push us. Okay, so what are some important features of this setup? What are some features that we want to know? So one feature we want to know is basically how far is the bacterium from the cell surface, right? That's a really natural thing that the cell will want to compute somehow, right? Because if it's really, really far away, probably it's sort of not worth its time to keep going at it, right? But if it's close, it will want to start actively engaging in the search process. So this distance r is something that the cell, we think, remember this is us just thinking about the problem, is something that the cell really wants to probably estimate, right? And so, okay, so the bacterium is shedding molecules. We're assuming that this neutrophil has receptors all over the cell. So what kind of process do you think will allow the shed molecules from the bacterium to find the neutrophil? Anyone want to give any guesses? Like, what happens to these ligand molecules that are coming off the bacterium? Yeah.
Passive diffusion.
That's a very good first guess, right? Let's just assume that the shed molecules undergo passive diffusion. Diffusion means, are you unfamiliar with the concept of diffusion? Okay, so what diffusion is it's essentially pure random motion due to thermal noise, right? So if you have a molecule, it's sitting in some solution, right? By random chance, water, say, molecules in that solution are going to bump that molecule, right? And the degree to which the bumping happens is completely random, right? So they'll come in from all angles, and they happen sort of at with time intervals that are also random, right? And if you think about what kind of motion that would give rise to, it's essentially like a random walker, right? So it's as if you're walking and you have equal probability of stepping one direction or another, because these water molecules are just coming in all different angles. Okay? And what's nice about that kind of motion is that it's a very natural, like, first guess. So this is us guessing what kind of motion these important molecules are going to undertake. Okay. So now, to take that idea one step further, I'm going to flash the diffusion equation. Okay, so this is the map. Now we've thought about the system. We've sort of built up a simple cartoon in our heads for what kind of processes we should try and keep into account. And now we want to do the step that oftentimes people get kind of worked up about. But I don't want this to be like too much of a shock or anything, right? So this is something that we will learn to do in this class. And at this step between posing the question, writing out a cartoon for what we think is going on, and then translating that into mathematics is literally what we're going to come back to over and over through this in the class. Okay, so the diffusion equation is actually not the easiest equation to start with. So it's like, it's a little bit complicated. But let's walk through all the terms, because anyone can understand what these terms are about. So what we're trying to do is we're trying to compute how this object, which is C, the concentration of the ligand, so the concentration of the molecule that is coming off the bacterium, how does that C change? How does that C? What is the C? What is the concentration as a function of how far away the bacteria is from the cell and time? Okay? And what this equation does for you is it tells you how that concentration changes with time. Okay? So that's the left-hand side. So in the kinds of equations we're going to usually be writing down, the left-hand side always encodes the object whose properties we're trying to study and how it changes with something else. Usually it's time. We just want to solve for how these molecules move in time, or or how some signal propagates with time. But sometimes it'll be other things. It'll be space. It'll be how the activity of one thing changes with respect to the activity of something else. We'll get into all those examples, but that's what the left-hand side tells you. It tells you basically what are you trying to study, right? The right-hand side tells you mathematically how different processes affect this change in time. Okay, so we're trying to study how the concentration changes the function of time, and what this you don't have to know right now at all. This we're going to return to in like week 12 or something, much later. But basically, what this tells you mathematically is that what we have is a random walker. It's something that undergoes random motion with equal steps left and right. Okay, now that's a purely physical process, right? That takes care of how we think these molecules from the bacterium arrive at the surface of the cell. But when it gets to the surface of the cell, I mean, absent anything else, okay, it'll get to the surface of the cell and then it'll bounce off. How is the cell actually going to sense that, right? So on that side, you have a different sort of almost description of even how to start, right? So while in the diffusive nature of ligand shedding from the bacteria, you have this pure, just this easy, or this sort of very simple equation to describe its motion. On the binding side, on the sensing side, for how the actual ligand will get sensed, we have a chemical reaction, basically. In this case, a very simple chemical reaction. But for those of you who have done sort of chemical kinetics or have done sort of freshman chemistry, this kind of thing should look pretty familiar to you. Anyone know what's going on with this reaction? Volunteer? What's happening?
Is it like the Chatelier's principle?
It's kind of like that, yeah, exactly. So basically, you have the chemoattractant, C, the ligand that the bacteria is shedding, plus R, which is the receptor. And what's happening is it's binding with some rate, right? So as a unit of time, there's a number of binding events that you can record, right? And it's the rate at which it's happening is called K on. Okay, that's just for simplicity. So basically, C and R get together, and at some rate, they'll stick to each other, right? And that rate's K on. And then similarly, in reverse, this is a reversible reaction. You can have this bound complex of chemoattractant and receptor that's stuck together, can fall apart, right? It can go back into its constituents. And that's the C plus R. And that happens at a rate K off. So this can also be just like we translated this picture using our brains to come up with the diffusion equation, there's even a more formal way to turn reaction schemes like this into sets of differential equations like this. Okay? And today, by the end of today, we'll go through one example of how to get that started. Okay? And that translation of chemical reactions that look like this into simple ordinary differential equations is something that we're going to do a lot of in the first half of the class. So what's nice is that you can use this combination of physical ideas, diffusion, and chemical ideas, ligand binding, receptor binding, to solve for something that it makes sense that the cell would want to monitor, right? Which is the fraction of receptors that are bound as a function of the distance, right? Because the idea is that if you combine these two processes in one framework, then you can imagine that the closer this thing is, the less diffusion will spread those chemoattractants around, and so the more these will be bound, right? The bigger fraction these receptors will be bound. So this is I'm not saying that we're going to sit here and figure out how this works. I'm just saying that this is the kind of thing that we can compute, right? We can compute things like what fraction of receptors are bound as a function of distance. We can also just compute what fraction are bound at an infinite distance. This is now a well-posed problem that we can actually analyze mathematically. You're going to learn all the tools you need to know to do exactly something like this. Okay. Yeah, so that's just words, what I just said. Okay, so let's go over quickly the sort of nitty-gritty mechanics of the class, just so you have an idea of what's coming down the pike at any given moment. So the class basically exists in two halves. The first half is one where we're going to treat the cell as a well-mixed system, meaning all that stuff I just talked about, diffusion and all that, ignore. Okay? We're just going to treat the cell as a well-mixed system, no diffusion, no gradients, nothing like that. And what we'll do is each week we'll step through a different set of, through a given topic. We'll devote the whole week to that one topic so that we really are able to apply the tools that you're going to learn to a given system. So to start, we're going to do a lot of what we did at the little end of that little toy exercise. We're going to use ideas from chemical kinetics. So we have rate equations, like how fast do things bind to each other, how do they unbind. And we'll apply that kind of thinking to simple models of gene expression and signaling. So for those of you who are more from the biology side of the world, I think we'll be very used to these kinds of, the vocabulary at least of what we're talking about. We're going to talk about things like, when you want a cell to make a certain amount of messenger RNA, how can you actually compute something like that based on the transcriptional regulation that the cell is employing in that particular system. So we're going to do a lot of this. Once we have a little bit of background in how to translate chemical reaction schemes into simple equations, then we can sort of start to analyze slightly more complicated systems, right? Things that have interesting qualitative features due to nonlinearities in their interactions. So one thing that is very true of biology is that biology is just rich with really, really nonlinear behaviors, meaning that you can't just say that the output of two processes is the sum of the two inputs, right? There's a lot of rich interactions that actually lead to much more interesting behaviors than what you get from a simple linear system. So for this, we'll do a little bit of nonlinear dynamics and apply some of these ideas to simple synthetic circuits that people have studied. And you've heard of like the genetic toggle switch or the recresilator or anything like that? Yeah. So that's the kind of system that we'll look at for that week. And then we'll, continuing on that theme, we'll look at things like how genetic oscillators work. So in particular, we'll pay attention to the cell cycle, which is the most fundamental oscillator we have, right? So that's how that really describes how cells divide. And then we'll start getting into we'll try to combine some of the ideas from the earlier weeks into more integrated systems. Things like bacterial chemotaxis, where different networks need to talk to each other in order to achieve the function that the cells are trying to achieve. And then we'll, so all of the stuff that had happened before will have all been with ordinary differential equations. So in the sense that the system is perfectly deterministic. So if I tell you the initial conditions that the cell is prepared in, and I tell you the equations that tell you how things change with time, you can tell me with 100% precision how, what the system will look like 10 minutes from now. Okay? That's a purely deterministic system, which if any of you have done any kind of biology experiments, you know is like manifestly like not true for a lot of things, right? Like biology is full of random behaviors, and so after we've mastered this deterministic approach to analyzing biological systems, we'll look at what happens when you add noise to the system, right? How does the cell generate noise? How does it deal with noise? How does it exploit noise? So this is a big theme in sort of modern quantitative biology research is how does this phenomenon of stochasticity interplay with biological systems to achieve functions. And then once we have this notion of probabilities, we can actually apply formal tools of probability theory. Namely, we'll look at what's called information theory as a way to describe and compute how these systems are performing. Okay, so that's the first half. The second half is all about adding space back into the system. So, whereas the first half is the cell is a well-bagged mix of reactions, in the second half we're going to start looking at spatial diffusion. So the first thing we'll do is we'll do a quick introduction into eukaryotic cell biology, because that's the scale where these spatial effects become even more pronounced. There are spatial effects even in bacteria, even in viruses, but it becomes really more manifest in eukaryotes. So the first thing we'll look at is something actually that is this phenomenon of the creation of subcellular structures, but that still largely ignore a lot of the spatial degrees of freedom. So sort of a nice transition between the two topics. So these are these so-called membrane-less organelles, which I don't know if any of you have worked in sort of biology labs, you know that basically you either work on this or you're dead to Google right now. Like this is a very hot topic of research right now, is the formation of organelles without membrane-bound, without membranes to bound their surfaces. And this occurs through really sort of classic phase transition kind of pictures that we'll go over. After that, we'll explicitly start including things like diffusion into the models that we're going to consider. And then once we have done that, we'll do an intro into what's called active matter. And active matter are systems that basically burn energy to create patterns on these spatial scales. And then we'll return again to the problem that we were starting with here, right? With things like, how does the cell actually combine all these different things, diffusion, chemical kinetics, and transport and cellular motility, to actually do things like chase bacteria. So we're going to introduce models about what's called gradient sensing. So this is a model by which cells can actually sense the differences in concentrations to tell that it's correctly chasing a bacterium. And then finally, we'll look at larger, more complicated systems. And here we'll get more qualitative and less sort of computing a lot of things with pen and paper. It'll be more looking at pattern formation and what are called Turing instability. So this is the problem of how do zebras get their stripes. And then finally, we'll do a little intro into ecology to round out topics. Okay. And then just some brief notes about how this, the nitty-gritty mechanics of the course. So we are going to do all homeworks in the class. And so these problems can be fairly challenging, and so I don't want time to be such an issue where you have to sit in the exam room and try to figure it out. So the homeworks will be every other week, so there'll be 6 of them. The first one will be due at the end of the 3rd week, and then they'll be sort of every 2 weeks after. They'll be the biggest part of the grade. It'll be just homework. And then for those of you taking 454, there will be a little bit extra for you to do. Usually it involves literature, searches, and stuff like that. See how these ideas actually play out in modern research. And then the last bit of the grade will be based on the final project. It'll be 20% of the grade. And I'm going to try to do something a little different this year, where because there are a fair number of you, I encourage you. You don't have to do this, but I encourage you to sort of pair up, right? So you can do the final project either. You can do it by yourself. You can do it in pairs. I think we will stop it at pairs probably. I don't know if you want to do triplets, then you really have to have something pretty spectacular probably. But I am persuadable. I'm always persuadable of these things. So, and then what I'd like to do is kind of do like a lightning round of proposals. So at the end of during week 8. The teams you guys will propose the idea for your final project and give like a 5-minute quick overview of what you're planning to do. And that way everyone gets to kind of see what everyone else is doing, because I think it's kind of fun. I mean, with a large enough population of people, you guys are going to come up with really amazing things, I'm sure. And then for the 454 students, it would be better for your own education if you didn't do just sort of reproducing an analysis of something that someone has done before. So do something a little bit more original. Okay. Any questions about it? You can come talk to me about that stuff anytime you want. That's fine. And I'll post all that on the website, so that it's more written out. All right. So before we wrap up for today, let's get some sort of real science under our belts. So let's start with how do we go about turning reaction schemes, like the thing we saw at the chemotaxis example, into simple equations that we can actually use. So the thing we're going to start with is sort of a very important problem that people actually had worked on for decades and didn't have good insight into how to even think about this problem, which is the problem of, let's say you have to turn some substrate, okay, some chemical, into a product with some enzyme. Okay, so the enzyme, all it does is it takes in the substrate and spits out a product. And people have been struggling with the problem for, like I said, decades. So who can tell me what the defining feature of an enzyme is before we go forward? Yeah.
Is it the active site?
That's an important chemical feature of the enzyme. What about from bookkeeping, like, what happens to the enzyme during a chemical reaction?
It's not consumed during the reaction and is reusable.
Exactly. So that's a very important thing. So enzymes don't get consumed during chemical reactions, right? They don't dissipate them. Okay. So let's write down the reaction scheme that corresponds to this, linguistic, this language-based description of what we just said. So what you have here is you have an enzyme. It reacts with a substrate. Okay? And the key insight that Michaelis and Menton made into this problem is that you don't go straight from this enzyme plus substrate into the P, the product. Okay? You actually stop at this intermediate state called an enzyme substrate complex, okay? A transition kind of state. And it's only this complex state that then drives forward the reaction toward making the product. Okay. This is not universally true of all chemical reactions, but this for enzyme reactions, which are a broad class of reactions that we're going to concern ourselves with in the class, this description is almost always our starting point, basically. Right? So this is like the basis from which we can add bells and whistles or take things away as needed, but this is often where our thinking is going to start. An enzyme reacts with a substrate, makes a complex at some rate K1. That complex will make the product at also some rate K2, but what's important is it can also fall apart with some rate K minus 1. And the enzyme is recycled back, right? So you don't lose it. So now what our job is we want to take this chemical reaction and turn it into simple differential equations that we can analyze. Okay. So, and the goal at the end of the day is actually to compute how fast does that P get made. So you can imagine lots of cellular systems wanting to monitor how fast the product of some enzyme is happening, right? So metabolism is a really good example. Okay, so let's look at this. So the first thing we can do is we can write down all the terms in this chemical reaction scheme that affect how E, bracket E, so the concentration of E, how that E changes with time. Okay? So what are the two things that can happen to E as a function of time? Yeah?
It can be consumed by making the enzyme substrate complex, or
Okay.
it can be broken. It can be produced by breaking apart of the enzyme substrate complex.
Very good. That's actually even more precise than what I was going for, which is good. So there are two things that happen. You can make the enzyme, you can make E, and you can destroy it, right? You could eliminate it. At a given moment in time. Remember, we're not getting rid of it from the system, but at a given moment in time, E, which is the free enzyme, right, not bound to substrate, you can make it, or you can take it away. That's it. And now what we can do is we can take that reaction scheme and figure out all the different ways you can make it and all the different ways you can take it away. So how are we going to make it? So there are two ways that we can make it. And so, yeah, what were the two again? How do you make it?
You can make it by breaking apart the enzyme substrate complex.
Okay, right. So this one.
Reversing that,
That's
and
a
then
K.
also
So what rate comes with that?
yeah, going backwards and then
Right.
going forwards of that.
Exactly.
So making
Very good. So K minus 1 will enter the picture here because that's one way you can regenerate just free enzyme from the complex. And free enzyme comes out from actually doing the thing that this enzyme's supposed to do, right? Make the product. So it's going to be a combination of this K1 and K2 processes. And then to destroy free enzyme, there's really just one way, right? You bind it. You bind the enzyme and substrate together into this enzyme substrate complex. Okay, so knowing that, let's now write down symbols corresponding to these different processes. So let's look at the creation terms. So the creation terms will be K minus 1 times the concentration of the enzyme substrate complex. So that's one channel, that's one way in which you generate free enzyme. And remember, it's a rate equation, so it's in units of per time. Okay. And the other way is you make it by doing the reaction proceeds as you type. Okay, and then for the destruction, it's very similar, right? You break apart this. That's wrong. Actually, this should be. No, that's right. Yeah. So you join them together, right? So you consume the enzyme putting it into the boundary complex. Okay, and then you can do the same thing for all the other species in the system, right? You play the same game of combining rate constants with the concentrations. And then at the end, you get this equation. The concentration of product as a function of time. That's what we actually want to solve for. And it turns out to be K2 times that concentration, the enzyme substrate concentration. Everyone with me? Okay? So this game of turning reaction schemes into these kinds of equations is what we're going to be doing for a bunch of these. Okay? And next time what we're going to do is we're going to actually solve this equation, okay, using simple algebra, actually, because we're going to assume that this is the steady state, so this time derivative over 0. So we'll just do algebra to actually tell us how this system is working. And that's what we're going to do next time. And then we'll do a little bit of justification for why this description is relevant. All right. So yeah, if you guys have any questions, please